Form 7, Biology, Energy Flow, Ecosystem and the Environment. Topic 5b, Ecology. Learning objectives. Students should be able to understand what is meant by the terms population, community, habitat and ecosystem. They should understand that the numbers and distribution of organisms in a habitat are controlled by biotic and abiotic factors. You should be able to understand how the concept of niche accounts for the distribution and abundance of organisms in a habitat. Students should get a clear idea about the core practical level. And finally, they should understand the stages of succession from colonization to the formation of a climax community. First of all, we should know that organisms doesn't exist on their own. They have complex relationships with other species in the habitat as well as their physical environment. So, ecology is the study of the interactions between organisms and their environment. So, in this lesson, there are some key words that we should know. We will start off with the term ecosystem. It includes all the living organisms in an area and all the non-living conditions as well. It's a combination of the living organisms and the physical environment associated with them. Ecosystems consist of a network of habitats and communities. What is a habitat? It's the place where an organism lives, such as a stream, a tropical rainforest or a sand dune, and also a very small area of habitat is known as a microhabitat. For example, a tiny drop of rainwater could be a habitat for many varieties of organisms. Population is all the organisms of one species in a habitat. For an example, we can take a population of ladybirds in a field. But when it comes to community, it's all the different species living in a habitat together at a particular time. For an example, a habitat such as a rock pool will include communities as seaweeds, shrimps, crabs and other crustaceans. The niche. A niche of an organism can be described as the role of the organism in the community or its way of life. To give you an illustrative example, let us place ourselves in the African savanna. Now you can see in this slide, you have a nice picture of the African savanna or the deserts. Giraffes and gazelles and dung beetles and other carnivorous animals all live in the same habitat in a savanna. But they all occupy their own niche. Giraffes and gazelles can live together because each species eats a different variety of plant and they are not in direct competition. Dung beetles lay eggs on the feces of the above mentioned herbivorous animals, and they feed on these feces, and eventually dung beetles helps to fertilize the soil of the environment, and it will help the growth of the plants in the environment, and in turn it will help to feed the herbivorous animals living in that particular environment. So we can think that each organism plays its own role in the ecosystem, fulfilling their own needs and helping others to coexist in the same ecosystem. The biodiversity and distribution of organisms within an ecosystem is due to both abiotic and biotic factors. Abiotic factors are the non-living elements of the habitat of an organism. They include those related to climate, such as the amount of sunlight, temperature, rainfall, the amount of pH or moisture in the soil, and they have a great influence on the success of an organism in a habitat. Biotic factors are the living elements of a habitat that affect the ability of a group of organisms to survive or thrive in that particular environment. For example, the number of suitable prey species will affect the number of predators in a habitat. Any kind of living factor which is going to affect on the living organisms is a biotic factor. So as an example, competition for environmental resources, predation, diseases or parasitism, food availability can be named. What is succession? Our world is constantly changing. New islands are being created. Can you remember recently there was an incident in Australia where a wildfire occurred and nearly 28 million acres of land area and vegetation was destroyed completely. So nothing remains in the same, and habitats are continuously evolving and changing. The major biomes of the earth have developed over millions of years from bare rock into the ecosystems of today. This is the ultimate result of succession. It's a process by which the communities of animals and plants over a certain period of time are replaced by other more different communities and more powerful communities. Ecological succession can be two types. The first one is primary succession, second one is secondary succession. We'll move on to primary succession. The definition is pretty straightforward. It's the establishment and development of an ecosystem in an uninhabited environment. In this process, the ecosystem is going to go through a series of changes. Primary succession always starts with an empty, inorganic surface or a bare rock. Mainly, there are few major reasons for these types of gradual changes occurring within an environment. So, as you can see in this picture, volcanic lava can create new land. An eruption of a volcano will release large amounts of lava, and by the time the lava will pile up and it will solidify and it will create a barren land. Another way of creating a new land is according to the primary succession process is a glacial retreat. In this picture here, you have a frozen glacier, and with the time, as the seasons change and the planet is also warming up, so this glacier retreats and exposes a new barren land which can then be developed into a luscious ecosystem. The first stage of primary succession is colonization, and the first organisms to appear on land because of this colonization process is known as the pioneer species. Pioneer species are the first organisms to inhabit this new land. For example, algae, mosses or lichens can be considered as pioneer species, as these organisms can penetrate the hard rock surface and spread themselves, resulting in a bare rock to break into small particles. Along with these organic material, organic humus will also develop, and together they form a new soil. As the new soil is formed, other species like ferns and grasses can establish their root systems inside the new soil, and eventually the new soil layer will develop into more layers. More water and nutrients will be available, and gradually other larger plants can also grow in this environment. So as you can see in this illustration, the primary succession starts from bare rock which came from an volcanic eruption, then it will pave the way for the lichens and mosses to spread themselves in the land, and gradually it will become a luscious environment. With the time, the thickness of the soil increases and more organic matter will also get accumulated in the soil. So, this situation will allow taller plants to grow. Because of the development of the vegetation, more habitats can occupy themselves in this vegetation. Eventually, a self-sustaining climax community will appear like this. Let's move on to secondary succession. It's a change that takes place after this disturbance occurred in an environment or an established ecosystem. A disturbance in the sense could be a wildfire or a flood, a hurricane, or even a harvesting process of a crop done by the influence of the humans. When it comes to secondary succession, it's much faster because the soil is still there. So the number of plants and animals present right at the beginning of this secondary succession stage is also comparatively higher. As you can see in the given illustration here, after few years, after a wildfire, the secondary succession process will re-establish the ecosystem. So we'll move on to the next slide. So here you can differentiate between primary succession and secondary succession. Primary succession occurs in the land where there is no initial vegetation, whereas the secondary succession occurs in a land that has primary vegetation. Primary succession can be initiated due to a biological factor or an external factor, but secondary succession always starts with an external factor. Surface soil cover is absent when it comes to primary succession, but in secondary succession there is a surface soil cover at the beginning. In primary succession, pioneer species come from outside of the environment, but in secondary succession, pioneer species develop partly from the existing environment. In primary succession, it needs more time to complete, but when it comes to secondary succession, it takes comparatively less time. As we move on further in this lesson, it's really important to understand how different factors affect living things and determine the distribution and population size of an organism in a particular habitat. Living communities in a habitat is controlled by both biotic and abiotic factors, no matter what stage of succession is going on. So, abiotic factors are light, temperature, wind, oxygen, availability, soil structure, and biotic factors can be predation, reproduction, parasitism and disease, territorial behavior. So, when it comes to light, plants are dependent on light for photosynthesis. And temperature, every organism has a range of temperature which it can grow successfully and reproduce. And the temperature of the environment affects the rates of enzyme control reactions in plants and ectothermic animals. Wind increases cooling and water loss from the body of an organism. When we consider the abiotic factor water, there are certain organisms rely on water currents for the food resource. In a terrestrial environment, the availability of water is affected by several factors such as the amount of precipitation, rate of evaporation, and water loss by drainage through soil, like that. So, when we look at the predation process, scientists found that relationship between a predator and prey population is an oscillating relationship, or in other words, it changes between two extremes as a repeating cycle. Because when a prey population increases, there is more food for predators, and eventually the predator population will grow, and it will reach to a point at which they are eating more prey than the reproduction levels of that particular prey. So the numbers of prey will fall. This will reduce the food supply for the predators, and predator population will also decrease slowly, paving the way for the prey population to increase once again in the environment. When it comes to parasitism and disease, these abiotic factors can have a devastating effect on individuals in a population. Diseased animals will become weak eventually and will not reproduce successfully, and they can't hunt well, so with the time it will create a life-threatening situation for these animals. The abiotic and biotic factors that affect the number of organisms occupying a particular niche may be density independent factors or density dependent factors. So what are density independent factors? These factors affects all population regardless of their size. But when it comes to density dependent factors, it occurs when population reaches a certain size in a given area. Density dependent factors can be competition, predation, parasitism and diseases, and density independent factors can be climate changes, droughts, unusual winters, hurricanes and natural disasters, seasonal cycles, and even human activities. Competition. Competition comes into play when resources are limited, and it's considered as a major evolutionary force, a part of the struggle to survive. If there is not enough resources to go around, those with the best adaptations, they get to have them. No two species can occupy the same niche, and competition occurs for abiotic resources as sunlight, minerals, water, and biotic resources such as reproductive mates. There are two types of competition: intraspecific competition, interspecific competition. Intraspecific competition is the competition between members of the same species within the same niche for the limited resources. Intraspecific competition is the competition between different species within the same niche for biotic and abiotic resources. When scientists are considering the ecology of an area of a natural habitat, they will measure both the abundance and the distribution of the organisms in that area. Abundance is the number of organisms in a species relative to the number of the other organisms in the same habitat, whereas the distribution of a species is how it's organized in a certain environment. We can collect data using a number of ecological techniques. The first type that we are going to consider is random sampling. Random sampling is usually carried out when the area under study is fairly uniform, very large, or there is limited time available. When using random sampling techniques, large numbers of samples are taken from different positions within the habitat. A cord rod frame is most often used for this type of sampling. The frame is placed on the ground and the animals or the plants inside it is counted or measured or collected depending on what the survey is for. This is done by many times at different points within the habitat to give a large number of different samples. In the simplest form of random sampling, the quadrant is thrown to fall at a random position within the site. However, this is usually unsatisfactory because a personal element inevitably enters into the throwing and it is not truly random. True randomness is always important when it comes to ecological investigations because statistics are widely used to process the result of sampling. A better method of random sampling is to map the area and then to lay a numbered grid over the map by using a computer program or maybe manually. And as you can see here, this is a numbered grid map of an area to be sampled. Then we could choose which squares we should sample in by using the random number table. So the simplest way to sample an area is a frame quadrant, and this method is really useful for plants and animals that do not move much. This method is known as a quantitative sampling method as it allows to quantify and measure the individual count of an organism in an area, or we can take a percentage count of that particular organism. But there are limitations of this method because a decision should be taken if it's to include or exclude a particular organism which falls on the margin of the quadrant. This is the next technique, point quadrant. It is a horizontal bar supported by 2 or 3 legs. Along the bar here, they have holes, and a long pin can be inserted through the holes. The organisms or the species that each pin touches are recorded. And this is a method which we can use to find the percentage cover of a particular species, and it's a useful method to investigate a dense vegetation. The next technique is transects. Mainly there are 2 types. The first one is line transects, second one is belt transects. Line transects can gather data about an area in a systematic way. A transect is not a random process. Using a transect is not a random process. You can stretch a graded rope or a tape between 2 points as shown in this picture and record every single individual, either it's a plant or an animal, that touches the tape in between the 2 positions. When dealing with a belt transect, you use 2 tapes and stretch them on the ground, and between the 2 tapes you can do your survey. You can use quadrants for systematic sampling if you place them along a tape to form a belt transect like this. We can use transects to investigate whether a change in the distribution of organisms is linked to a change in an abiotic factor. It is important to be able to measure abiotic factors which may affect the abundance and distribution of an organism. You will use some of the techniques or methods that you can carry out in practical investigations to find the abundance of an organism in an ecosystem. This table here gives a summarized idea of the techniques that are commonly used and the instruments that are commonly used to measure abiotic factors. Temperature can be measured by a digital or a manual thermometer. Thermometer. Light intensity can be measured by a light meter. pH meter can measure pH values. Slope can be measured by a clinometer. Oxygen levels can be measured by digital oxygen probes. Wind speed can be measured by anemometers. Now we'll move on to another section of this lesson, statistics and ecology. So under that we have to discuss the co-practical number 11. It is a study of the ecology of a habitat using a quadrant or a transect. We can find the distribution and abundance of an organism by using these techniques. So here we are using an interrupted bell transect to investigate the effect of one abiotic factor. In this case, it's light intensity. We can choose a site where there is an obvious gradient in the abiotic factor, which is light. In this case, a shaded area under a tree canopy or the shaded side of a tall building to an area in full sunlight can be selected. We have to lay 20-meter tape as a transect line with the 0-meter mark in full shade here and 20-meter mark in the full light. Then we walk along the transect and decide whether we are going to pursue a percentage cover or just a count of the organism. Starting at one end at the transect, we lay a quadrant next to the 0-meter mark on the tape and record the abundance of the species that we selected and measure the light intensity at the ground level. Then move the quadrant 2 meters along the tape. Likewise, you can repeat the process, and if you want, you can take same measurements again and again. Remember that the distribution of organisms is usually determined by a combination of abiotic and biotic factors. So when we study the effect of one factor, all other factors should ideally be controlled. This is not possible when you go to a field and do the studies, but we have to make appropriate steps to at least try to keep the other variables as controlled as possible. So now we have done a field study. You will collect a lot of data. But it's not always easy to understand the data. It's hard to know whether patterns that appear are the result of biological relationships or it is due to some sort of a chance. This is why we have to use statistical tests to prove and decide. Before you start to collect data in the field, it is important to have a clear idea of what you are trying to prove or disapprove. In other words, you need a clearly stated null hypothesis, and this allows you to choose the right statistical test to help you to analyze and evaluate your data. After running your chosen statistical test, you will obtain a number known as the observed value. You need to look up this value in a published table of critical values to see which probability value this corresponds to. So you can decide whether or not your results are significant or whether you should accept your null hypothesis. In other words, if your results are significant, they are not due to a chance. If your observed results fits with the critical value corresponding to the probability value less than 0.5 level of the significance given in the table, you can be 90%, 95% sure or certain that the relationship between the factors you are studying is due to something other than chance. So scientists use statistical computer programs to run these tests and to give the probability value. So there are few types of statistical tests that we are going to discuss. The first test type is Spearman's rank correlation test. This is a statistical test used to check whether there is a significant relationship between two sets of data. This test can be used for instances where there are at least 10 pairs of data. As in most statistical tests, as the first step we have to do in this test is to state the null hypothesis. So the null hypothesis is there is no significant relationship between the two variables that we are considering. As step 2, we are looking for a correlation between variables we are going to compare and see these variables affects one another. So as the formula I have given you here, RS is the correlation coefficient. RS equals to 1 minus 6 times sum of d squared over n cubed minus n. RS is the correlation coefficient. D is the difference between the ranks. N is the number of pairs of our measurement. Step 3. After calculating an RS value, we have to compare the value of RS and with the critical value at a confidence level of 95%. And if the RS value equals or it's above the critical value, then we can reject our null hypothesis, and if RS is less than the critical value, we can accept our null hypothesis. Now we'll move on to a question. Here, a scientist wanted to know if there was a correlation between the heights of men and their sons. The heights from 11 father-son pairs are shown in the first two columns here. Now in this question they have given you the heights of the fathers as well as the heights of the sons. First step, we have to state a null hypothesis. There is no correlation between fathers and sons' height. As the next step, we have to rank the data given in these two columns. Ranking in the sense arranging them in ascending order from the highest value to the lowest or the smallest value. So here the highest value is 1.8 meters. So we gave it the number 1. Then 1.77 meters, it is the second highest. Likewise, you have to arrange or give numbers from the highest to the lowest value in considering both sets of data. So let's say while doing ranking, we found two equal values. So 1.77, there are two values here. So we don't give the same ranking to these numbers. What we do is we go through the ordinary ranking process, and finally we have to average the ranks out. Now here 1.77, this is the second rank and this is the third rank. So in between 2 and 3 you have 2.5. So we give these numbers 2.5 here. So likewise, I have conducted the same thing in this column also. 1.65, we have two numbers, two equal numbers. So the ranks are 9 and 10. You have to give a number in between the two values. So it's 9.5. So 9.5. Then as the third step, you have to find the differences between the ranks. So what is the difference here? 6 minus 4. It's 2. And we have to take the square root of that particular number. 2 into 2 is 4. So we'll move on to the next line. 8 minus 4 is 4. 4 into 4 is 16. So likewise, we have to complete this row and take the square differences, the sum of the square differences in this column. So it's 49.5. Now we can plug all the values to the equation like this, and you get a positive value for RS. It's 0.775. So there should be a positive correlation between the two data sets. So then now by using a table for the critical values for the Spearman's rank correlation coefficient, we can find the critical value at the 0.05 confidence level. It's 0.618, and we compare the critical value and the calculated value. So in this case, the calculated value is high, so the probability is less than 0.05, that the observed correlation is just due to a chance. So we can reject the null hypothesis in this case, and there is a significant positive correlation between father's and son's heights because we got a positive correlation. The next type of test is the student's t-test. The student's t-test is used to determine whether a mean of a variable in one group differs significantly from the mean of the same variable in different group. This test considers the degree of overlap between the two sets of data and allows you to judge whether any difference between the mean values of the two groups is statistically significant or just due to chance. To continue on with this test, we have to find the mean values of the two sets of data, the standard deviations of the two sets of data. By using the standard deviation, we can find the variance of the two sets separately, and we can plug these values into this equation. And there's another thing that you have to find. It's the degrees of freedom. It is equal to the sum of the sample sizes of both groups minus 2. So find the probability value that relates to the observed value and the degrees of freedom. If the value is less than 0.05, the difference between the means of the two groups is considered to be statistically significant. The third type of test is the chi-squared test. It is a relatively simple statistical method which we can use to estimate the probability that any differences between the observed and expected results are due to a chance. The chi-squared test will tell you whether or not your results differ significantly from the expected outcome. And this is the equation that you have to use in this test. E here is the expected value, O is the observed value of the frequencies. First, we have to state a null hypothesis. So we'll move on to a question. In one of Mendel's experiments with pea plants, he observed the flower colors of 920 offsprings from a cross between two heterozygous red flowered plants and observed 705 red flowers and 224 white flowers. From his theory, he expected a ratio of 3 to 1. Do these results agree with his theory? Let's have a look. So first you have to prepare a table like this. In this column you have to state the hypothesis or the ratio of the expected values, and you have to state the observed results and the expected results, and we have to take the difference between the observed and the expected results, and you have to take the square root of it, and you should divide it by the expected result value. And for after completing this row, you have to take the sum of these values. So in this case it's 0.387, and we have to calculate the degree of freedom. The number of categories is 2, and you have to omit 1, and 2 minus 1 is 1. So the degree of freedom is 1. So we refer to a chi-square distribution table. Degrees of freedom is 1, and according to the 0.05 confidence level, the value taken from the chi-square distribution table is 3.84. So we have to compare these calculated value and the critical value. So in this case, the probability is greater than 0.05, and the observed difference between the observed frequencies and a ratio of 3:1 is just due to chance. So we have to accept the null hypothesis. There is no significant difference between the observed frequencies and a ratio of 3:1. So try the exam practice questions on page number 48 and 49 in your textbook. Finally, we have come to an end of this lesson. In this lesson, we discussed what is meant by the terms population, community, habitat and ecosystem. And we discussed about biotic and abiotic factors and how it controls the distribution of organisms in a habitat. We understand how the concept of niche accounts for the distribution and abundance of a particular organism. We discussed about succession and type of successions. We discussed the differences between density dependent and density independent factors. We discussed about competition, sampling techniques, and how to measure abiotic factors. We got an idea of co-practical number 11, and we did statistical tests in the end of the lesson. So these are the references that you can go through, and hope you have enjoyed the lesson, and thank you.